Hello, today I will be presenting a project on high energy bonds for our CHEM 141 video project. So to begin with some background, the transfer of energy across high energy molecules is essential in biological life. It is required for the work for organisms to stay alive, grow, and reproduce. In metabolic pathways specifically, high energy bonds provide the chemical energy that fuels the synthesis of complex biological molecules from simple constituents. Other examples of the roles of chemical energy include the production of concentration gradients and electrical gradients in the cells, which are important for biological transport, as well as the production of heat and motion. And in some organisms, chemical energy is even used for the production of bioluminescence. Importantly, biological energy transfers obey the laws of thermodynamics. The main thermodynamic quantities to consider in chemical reactions observed in biological systems are enthalpy, entropy, and free energy. These quantities can be related to one another by the equation delta G is equal to delta H minus T, which is temperature, times change in S. Ultimately, the change in free energy is the driving force of chemical reactions, with chemical reactions proceeding spontaneously in the forward direction when delta G is large and negative. Cells, however, do not normally operate under standard conditions and therefore cannot follow that previous equation. In reality, the free energy of a reaction depends on both standard transform energy, free energy of change, which is delta G prime degree, as well as the concentration of products and reactants in a given reaction. Thus, a more accurate free energy change equation is shown boxed off in the red. From this, you can see that reactions are more favorable when more stable products are formed as related to H, and when initial concentration of reactants is high relative to that of the concentration of products, as well as at higher temperatures. Unfortunately, limitations to the reaction favorability exist in that cells have limited solvent capacity due to their small size and can only operate under a narrow temperature range. In order to overcome these limitations, activated intermediates can be used to push the transfer of groups in a chemical reaction at lower concentrations of reactants in order for reactions to proceed. Activated carriers are the molecules that act as these intermediates. These molecules function to transfer one functional group to another molecule, or one functional group from one molecule to another in order to move a reaction forward. They are said to be activated because the transfer of the specific functional group is energetically favorable. Phosphoryl group transfer is one such example of this. The transfer of phosphoryl groups is energetically favorable due to resonance stabilization of phosphate groups upon hydrolysis, as well as the inherent stability of the tetrahedral structure of phosphate. Thus, reactions that result in the cleavage of a phosphate group from one molecule to another, such as hydrolysis reactions, are made more favorable by forming the stable inorganic phosphate group product. Importantly, the energy associated with the cleavage of phosphate from an activated carrier is important to couple with for other inorganic reactions within the cell. ATP is the chief activated carrier of phosphoryl groups with an extremely favorable free energy of hydrolysis. Several factors contribute to the large free energy change associated with ATP hydrolysis. For one, hydrolysis results in the breaking of the high energy phosphoanhydride bond. This alleviates the electrostatic repulsion of the four negative charges on the ATP molecule. Secondly, as shown on the previous slide, the inorganic phosphate product is stabilized by resonance stabilization, making the formation of products favorable due to the stability of products compared to the reactant. And additionally, the ADP product produced by ATP hydrolysis is able to immediately ionize, which results in the favorable release of a proton in a very low proton environment within the cell, further increasing the favorability of product formation. These factors make ATP a metastable product, as it is itself thermodynamically unstable due to its charge repulsion, yet its reaction is extremely kinetically favorable and stable. There are other phosphorylated compounds besides ATP that also exhibit high free energy changes of hydrolysis. The hydrolysis of phosphoenol pyruvate, for example, results in the very high delta G of hydrolysis of negative 61.9 kilojoules per mole. This large free energy of hydrolysis is due to the fact that the reaction yields an enol form product of pyruvate that can immediately tautomerize to the more stable keto form. Thus, the product has two forms and is stabilized relative to the reactant, which only has one form. This makes the reaction very favorable. 1,3-bisphosphoglycerate is another high energy molecule whose hydrolysis is favorable. This is again explained by the hydrolysis reaction resulting in the formation of more stable products relative to the reactants. In this case, the ultimate product is stabilized after undergoing ionization, which allows the product to form two equal resonance structures. A final example of a phosphorylated compound with a high delta G of hydrolysis is phosphocreatine. Hydrolysis of phosphocreatine breaks a bond between phosphorus and nitrogen, which generates a free creatine that is stabilized by the formation of resonance hybrids. There are other molecules that have large and negative standard free energy of hydrolysis. One such example are thioesters. These contain a sulfur atom in the position occupied by an oxygen atom in oxygen esters. Their hydrolysis produces a carboxylic acid that can ionize and form several resonance structures. This overall reaction from unstable reactants to resonance stabilized products results in a large and negative standard free energy of hydrolysis. And shown on the right is the hydrolysis of acetyl-CoA. Activation of the acyl groups of acetyl-CoA is very important to transacylation, condensation, and oxidation reduction reactions. And as mentioned, the coupling of ATP transfer in biological processes is very important in metabolic pathways specifically. In glycolysis, for example, ATP acts as an activated carrier by transferring its phosphoryl group 2 different times. These transfers occur during the preparatory stage in which energy is invested. This investment initiates a series of reactions that ultimately yields a greater amount of ATP, as well as important biomolecules for later stages of metabolism. The specific reactions that ATP transfer in the preparatory stage of glycolysis play a crucial role in the continuation of the process. The first phosphorylation of glucose acts to trap glucose in the cell by giving it a negative charge, while the second phosphorylation by PFK1 is highly thermodynamically stable or thermodynamically favorable, thus making it the first committed and rate limiting step of glycolysis. And here are my 2 example exam questions as well as their answers. Thank you.